Welcome back, everyone. So this week we're going to get into molecular biology, so chapter 8. So I believe this would be chapter 13 in your Campbell biology book, if I recall. So in chapter 8, we have it divided into 5 different sections. So last week we looked into patterns of inheritance. So we looked at, how we inherit 2 copies of every gene from our parents. But we haven't really talked about, DNA itself. So, we talked about like the genes, but, the genes are made of DNA. So, think about genes. Genes, that DNA is just a sequence of nucleotides, and those nucleotides, there's just 4 of them. There's adenine, the A's and the T's and the G's and the C's. So we're going to focus in on those 4 nucleotides and how just a sequence of those will code for the proteins that code for our traits. And so we're going to start out by looking at the structure of DNA. And from there, we're going to learn about how DNA is replicated, and then how we can take that sequence of DNA and create an actual trait from it. Okay, and then we'll start getting into how we can regulate genes. Okay. So by the end of this chapter, we're going to, you should be able to describe the structure of DNA. And then you should also be able to describe how eukaryotic and prokaryotic DNA is arranged in the cell. So if you recall from earlier in the term, way back in, I think it was chapter 2, we learned that the macromolecule nucleic acid, DNA, is a polymer of nucleotides. So each of these nucleotides is composed of 3 parts: the nitrogenous base, a 5-carbon sugar, which in DNA it's a deoxyribose, and then a phosphate group. And then when we focus in on the nitrogenous bases, there's 4 of them. So we have adenine and guanine, cytosine and thymine. So, we're going to talk more about that structure. And then we're going to focus in on DNA being double-stranded, but we'll also look a little bit about how RNA is single-stranded, and we'll see the difference between the 2. Okay. So let's start out by looking at a little bit of history, so the discovery of DNA. So in the 1950s, we had a guy named Francis Crick and a guy named James Watson. They worked together in England. I think it was the University of Cambridge. They worked together to figure out the structure of DNA, but they weren't the only ones involved. There are a lot of people working on figuring out what DNA was. So they were just kind of the ones that were involved in putting the pieces of the puzzle together. But there are other people that were working on the pieces to begin with. So Pauling, Linus Pauling, he, along with Maurice Wilkins, they were also working on the pieces of the puzzle. So Pauling, he discovered the proteins that were involved with DNA. So through crystallography, X-ray crystallography specifically. So you can see here's like a structure involved with there. Wilkins, in Wilkins' lab, there was a researcher, Rosalind Franklin. She used the X-ray crystallography to understand the actual structure of DNA. So this was the piece of the puzzle that was missing for quite a long time, is to figure out how DNA was, this double helix structure. And she figured it out. So based on that data, Watson and Crick was able to use Rosalind Franklin's data to piece together the puzzle of DNA. Okay, and so based on that, they were able to publish basically the structure of DNA. They're, basically that it's a double helix, and that that DNA, the DNA molecule has the 4 nucleotides present, and that each of the nucleotides, if there's adenine, there's equal parts of thymine in it, and then if there's guanine, there's equal parts of cytosine in it. So that's something called Chargaff's rule, and we'll talk about that a little bit later. Based on that, in 1962, Watson, Crick, and Wilkins were all awarded the Nobel Prize in Medicine. So you might be wondering why Rosalind Franklin didn't receive the Nobel Prize, especially since her work was so critical in Watson and Crick, finalizing the structure of DNA. Well, she died in 1958, and so based on the rules of the Nobel Prize at the time, they couldn't award it posthumously, or which means that you can't give it to somebody who's already died, and so she was left off. I think they changed the rules like in the 90s or something, but that was obviously later. But she would have been awarded it if she was alive, but she had died before 1962 when it was awarded. So, so that's who discovered it. Usually Watson and Crick, you think of them as the main guys, but there were actually quite a few people that were involved. Okay, so we're going to watch a video describing the structure of both DNA and RNA, and then we're going to get into the details, okay? So let's take a look.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they could start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing 1 strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic, apples in the tree, that helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discuss transfer RNA, more abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles, but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATTGAC, can you complete what the complementary DNA bases would be for the other DNA strand? The answer: so remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer: so remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a brief introduction into basically what DNA is and RNA. So now we're going to get into the details. So like the video talked about, our DNA is made of the monomers nucleotides. Okay, so those are the subunits. And like the video talked about, the subunit nucleotide is made of 3 components. So we have the sugar, the phosphate, and the nitrogenous base. And you can see here in this diagram here, this figure, and this is from the textbook, that the sugar group is in the middle, and then we have the nitrogenous base, and then we have the phosphate group on either side, okay? And you can see that the sugar in DNA is deoxyribose, and that's a 5-carbon sugar, okay? And so if I was to label this, we would basically count the carbons, and we'll do this a little bit later. You'll see how it's counted, but we start at the oxygen and we go clockwise. Okay, so this would be the first carbon, the second carbon, the third carbon, the fourth carbon, and then the 5th carbon's up here. Okay, and you can see that in this case, it's actually, this isn't deoxyribose. This is this one's actually ribose for some reason. I don't know why they put an RNA sugar here. Wow, that's really interesting. This is RNA because deoxyribose doesn't have this OH here. It's just an H. But anyway, so hey, this is RNA, an RNA nucleotide. But yeah, it will have a ribose sugar just like a type of 5-carbon sugar, just like our RNA, okay? And then we have the phosphate group, so you can see there's a phosphorus with 4 oxygens here, and then a nitrogenous base, so you can see the nitrogens here, okay? And then if we look at our nitrogenous base, there are 4 types that are possible bases that attach to the sugar group for DNA. So that would be adenine, guanine, cytosine, and thymine. Okay, and if you notice, there's a couple differences here. Okay, so adenine and guanine, what do you notice that's different between adenine and guanine compared to cytosine and thymine? So cytosine and thymine, they're both single rings, whereas adenine and guanine are double rings. So we call adenine and guanine purines, whereas cytosine and thymine are pyrimidines, okay? So just realize that adenine and guanine are purines, cytosine and thymine are pyrimidines, okay? And this comes into play when we do our base pairings, okay? So when we do our base pairings, adenine always pairs with thymine, and guanine always pairs with cytosine. So we always have a purine pairing with a pyrimidine, okay? And it happens that adenine always pairs with thymine. The purine guanine always pairs with the pyrimidine cytosine. So that makes sense because we want it balanced out. So we have a double ring adenine pairing with a single ring pyrimidine. Okay, or thymine. All right. Now, one thing that's different between DNA and RNA, DNA is a double helical structure. So when we look at DNA, we have 2 DNA molecules, okay? So it's 2 strands of nucleotides. And when we look at it, the sugar and the phosphate groups form the backbone, okay? So this line right here, that is the sugar and the phosphate group, all right? So that forms like the sides of the ladder here. So that means that the part that's sticking out, kind of like, you know, the rungs of the ladder that you step on when you're stepping on a ladder, that's the nitrogenous bases. Okay, that's what's sticking out. And so when we have an adenine sticking out from one DNA strand and a thymine sticking out from another DNA strand, they will form a temporary bond with each other. And that temporary bond is a hydrogen bond. And if you remember, hydrogen bonds, those are weak bonds. Okay, so back when we learned about chemical bonds, covalent bonds, they're really strong bonds. That's what holds the sugar phosphate backbone together. The bonds that bring together the base pairs, those are hydrogen bonds. So that's a weak bond, okay? So that's why they're not really, you know, you can see them kind of coming apart. Sometimes they're held together. So the sides of the ladder, those are strong covalent bonds. The rungs of the ladder are held together by weak hydrogen bonds. Okay? All right, so let's take a closer look. So here imagine we have our double helix. So here's another look at it, okay? So here again is the side of the ladder, and that's our sugar phosphate backbone. So if we look at our sugar phosphate backbone, it's this part. And that's composed of our sugar and our phosphate of one nucleotide attached to the sugar and phosphate of another nucleotide. And you can see where it's attached. So we have the phosphate group of one nucleotide attached to the sugar of another nucleotide. And this attachment between the phosphate group of one nucleotide and the sugar group of another is a covalent bond. Okay, so this is a strong bond. Okay, so this is not a weak bond. This is a strong bond. Whereas if you take a look over here, here's the nitrogenous base. And this nitrogenous base happens to be thymine. And what does thymine bond with? It bonds with adenine on another DNA strand. Okay, so here's, one strand, here's the other one. So we have adenine here binding with thymine. Okay, so this thymine is binding with adenine, and notice it's just a weak hydrogen bond. Okay, so it's just a temporary hydrogen bond. Here's guanine forming a bond with cytosine. And it's just a temporary bond. All right, so the sugar phosphate backbone is strong. The base complementary base pairs between the nitrogenous bases, they're forming hydrogen bonds, which are weak bonds. Okay. Another thing that you'll notice is the arrangement of the strands. Okay, so look at where the sugars are facing in this strand. The O's are facing up. In this strand, they're facing down. Okay, so they're running anti-parallel to each other. So again, if I count the carbons, we start at the O here. So this is carbon 1, carbon 2, carbon 3. So just remember 3, carbon 4, and carbon 5. Okay, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So that's why we call this the 5' end. So this is the 5 carbon end. This is the 3 carbon end. Right? So we're labeling this particular DNA strand the 3' end and the 5' end. Okay. All right, so the 3' end and the 5' end. This one over here, we did the same thing, going clockwise. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So that's why that's 3' there. Down here, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. That's down here. Okay, so this one's going from 3' to 5', the opposite direction. Okay, so that's why it's going 3' to 5'. So this particular strand is going from 3' to 5'. This strand is going the opposite direction. So the two strands are running anti-parallel to each other. So pay attention to that because it'll help you in the next video when we get into DNA replication. Okay, so pay attention to the 3' and 5' ends. All right. All right, so now let's get into the structure of RNA. So RNA stands for ribonucleic acid, which means we have ribose sugar instead of deoxyribose sugar. And remember a couple slides ago, oh, look at that. That's a ribose sugar, right? So this is RNA, okay? Because we have our ribose sugar here, and then this is deoxyribose. So, yep, there is an error there. Okay, so here's a deoxyribose sugar because we removed an oxygen group. Okay, so this is what deoxyribose looks like, a type of sugar, versus ribose. Ribose has an extra oxygen group. Okay, but they're both 5-carbon sugars. So there's 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. All right. With the RNA structure, it's basically the same structure as DNA. Okay, we have, a 5-carbon sugar, just like we had with DNA, just slightly different. We have a phosphate group attached to, the 5th carbon. And then we have a nitrogenous base attached to the 1st carbon, just like in DNA. The only real difference is, again, the type of sugar. And instead of thymine, there's something called uracil instead. Okay, so it could have adenine there, it could have guanine there, it could have cytosine, but instead of thymine, there's uracil. All right. So the nucleotides are very similar otherwise. Okay, so they have phosphate, they have a nitrogenous base. It's just they have ribose sugar instead of deoxyribose, and then they have uracil instead of thymine. The other difference between DNA and RNA, DNA tends to be double-stranded. RNA is always single-stranded. Okay, so you won't see 2 RNA strands next to each other. All right, as a double-stranded. So RNA is single-stranded. All right. But you will see RNA build off of DNA, like we'll talk about in the next video with DNA replication. Okay, so adenine will bind with thymine on DNA. Guanine will bind with cytosine on DNA. Cytosine will bind with guanine on DNA. And uracil will bind with adenine on DNA. Okay, it's just there's no thymine on RNA. So if there's an adenine on DNA, it will bind with uracil on RNA. Okay. All right, so that's the difference between DNA and RNA. So let's get into how DNA is arranged in the cell. Now, DNA can be very, very long. So for humans, our DNA, if we took all of our 46 chromosomes and spread them out, from end to end to end, it could be 2 meters in length. Okay, 2 meters in length for our DNA, that's a lot. Okay, so that's pretty long. So how do we get our DNA that's 2 meters in length in a tiny itty bitty cell that we can't even see with the naked eye, in the nucleus of the cell, let alone. Well, for one thing, our DNA is housed on linear chromosomes, and so that's, we'll learn about how it's kind of condensed in that. For prokaryotes, think about prokaryotes. Prokaryotes, their DNA can be just as long as ours, like E. coli, for example, the stuff the bacterial cells that can be in our gut that can cause disease sometimes. They can be 1.6 meters in length. They're just a single circular chromosome. Okay, so prokaryotes, like bacterial DNA, it just comes in a single circle. Okay, so single circular DNA, that's in the nucleoid region of the prokaryote. All right, so it's just kind of hanging out there. Eukaryotes, we are in, our DNA is found in the nucleus, and it's housed on linear chromosomes. Okay, so for us humans, we have 46 chromosomes total. Different species have different numbers depending on the species. Some species have more than enough, some species have less. It doesn't matter how many. Like, plants tend to have a lot more than us. Some of them can have like 5,000 chromosomes. Doesn't mean that they're more intelligent than us, so the number of chromosomes don't relate to anything. Okay? So that's housed in the nucleus. So how do we get so much DNA crammed in such a tiny space? Well, we gotta condense it, okay? So I like to think of DNA as like a thin piece of thread. And then think of histones, which are little proteins, as like a spool. So, if you unravel thread, it just creates like a big clump, right? That's big. But if you, start to wind it around a spool, it becomes smaller. And so DNA is wrapped around these proteins or histones to form a structure called a nucleosome. Okay, so here's like a little protein, and the DNA is wrapped around it. So it starts to form like these beads on a string. So these little yellow things are the protein, and then the DNA is kind of wrapped around it. And then it forms these little beads on a string. Okay, and then as the nucleosomes are formed, if it's just an interphase, they just stay like that. Okay, so they're a little bit less condensed. They're just kind of spread out more during interphase. But as we know in mitosis, under a microscope, we can actually see the chromosomes. So the nucleosomes are going to start to further stack on each other. Okay, so they're going to further condense. So they're stacking on each other and becoming tightly coiled around each other to form the actual chromosome that's visible. All right. So you can see how, what it looks like when it's organized. So, we have our regular DNA that's just kind of spread out, and then it starts to coil around a histone. So here's our histone. So it starts to wrap around the histone to form the nucleosomes. The nucleosomes, we have these kind of beads on a string that start to kind of condense. And then they're condensing further to form kind of a condensed chromosome that we can see under a microscope. All right. Okay, so we're going to end with just a video. This is from your textbook, so you can kind of see.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, So again, that's kind of cool. You get to see the different, not only the different stages from mitosis. You saw, hey, there's, prophase and metaphase and anaphase and telophase. But you saw how the DNA, started coiling around the histone to form the nucleosome. And then the nucleosome started coiling even further to form the chromatin. And then you saw it like condense even further so that, you have this structured chromosome that you could visually see under the microscope. Okay. So let's do some review questions and then we're out of here. So which of the following does cytosine pair with? Is it adenine, guanine, thymine, or a pyrimidine? If you chose guanine, I would agree with you. Remember, cytosine, car in the garage. So cytosine and guanine. All right, so let's do this review question. Prokaryotes contain a blank chromosome and eukaryotes contain blank chromosomes. So is it A, prokaryotes contain a single-stranded circular chromosome and eukaryotes contain single-stranded linear chromosomes? Or is it B, prokaryotes contain a single-stranded linear chromosome and eukaryotes contain single-stranded circular chromosomes? Or is it C, prokaryotes contain a double-stranded circular chromosome and eukaryotes contain double-stranded linear chromosomes? Or is it D, prokaryotes contain double-stranded linear chromosome and eukaryotes contain double-stranded circular chromosomes? So how do we fill in the blanks? If you chose C, I would agree with you. So prokaryotes contain a double-stranded circular chromosome. So remember, all DNA is double-stranded. And for prokaryotes, it's circular. And eukaryotes contain their linear chromosomes. Okay, so C is the correct answer. RNA is single-stranded, DNA is double-stranded. Prokaryotes have circular chromosomes, eukaryotes have linear, so C is the correct answer. All right, so we are done with our first segment of Chapter 8, Molecular Biology. So we looked at the structure of DNA. By now you should be able to describe the structure of DNA, as well as describe how eukaryotic and prokaryotic DNA is arranged in the cell. So for the next video, we're going to get into how DNA is replicated in 8.2.